Thank you very much for the kind introduction. So one of the things my lab's been doing for about the past decade is to use functional genomic approaches to try to identify new genes and regulatory pathways that promote or prevent cancer. And what I thought I'd do today is tell you three stories that use that type of approach. Here are the three topics that I'll cover today. I'll talk about oncoprotein-directed tumor suppressor gene silencing. I'll tell you about how an oncogenic splicing factor works. And I'll talk about a project that started a long time ago when Narendra was first in the lab on oncoprotein-directed senescence and apoptosis. Now, all three of these are in common in that there's a pathway that's initiated by an oncoprotein that affects cancer, and that we use some type of functional genomics approach to identify the pathway that's involved. In other ways, they're very different. This is a very mature subject matter, and I'll mostly be telling you about the lessons we've learned. This is a relatively new project. All the work I'll be showing you is unpublished, and this started a long time ago, but most of the work I'll be showing you today is new work. So let's begin with the first subject. When tumor suppressors were first identified, they were found to be inactivated by mutations in the protein coding region or deletions. But over the past several decades, it's been appreciated that a very common way that tumor suppressor genes become inactivated is they become transcriptionally repressed by a mechanism called epigenetic silencing. The epigenetically silenced tumor suppressor gene has the hallmarks of heterochromatin, hypermethylated DNA regions, and repressive chromatin marks. Now, important difference between these two modes of inactivation is while this is irreversible, this can be reversed, and there's a lot of interest in the biotech and pharmaceutical industry about finding biological or small molecule inhibitors that can reverse silence and reverse epigenetic silencing and restore tumor suppressor gene function. Now, what was unclear and what got us into this subject matter, I should say that in addition to single genes, there are also cases in which you can classify classify cancers based upon this widespread methylation phenotype called the CpG island methylator phenotype. And for example, colorectal cancers where this widespread aberrant silencing was first identified, you can cluster them into three types: CIMP negative, CIMP low, and CIMP high. And this CIMP phenotype has now been found in a lot of other cancers as well. What was unclear when we started this work was how the gene became silenced during cancer development. And there were two competing models. One of them, illustrated over here, was that acquisitions of repressive marks that would result in silencing occur randomly and then get selected for because this confers a growth advantage. And a second model which posits that there's a defined pathway starting from the oncoprotein. And we thought we could distinguish between these two possibilities by performing loss of function screening to identify what factors might be involved. So we've developed a number of methods by which we can screen for factors involved in epigenetic silencing of tumor suppressor gene. This shows one over here in which we would take a tumor suppressor that's inactivated in a particular cell type. We take the promoter of that tumor suppressor, we fuse it to a selectable marker like a blastocytin reporter gene. We put this into cells in which the endogenous tumor suppressor is silenced. And then we screen with shRNA library pools for reactivation of this gene. And when the gene becomes reactivated, that means that it's because this shRNA is directed against a component in the pathway. And so doing that, we've identified factors that mediate epigenetic silencing of tumor suppressor genes in cancer cells. We can then order these into pathways using epistasis analysis. And we find in all cases we've delineated pathways that are initiated by the oncoprotein and they culminate in silencing of tumor suppressor genes. And this just shows some of the examples of the pathways that we've identified. They all have in common several features. They're all initiated by an oncoprotein. They all involve a sequence-specific DNA binding protein, which confers specificity on the promoter, and they all involve a DNA methyltransferase, which is responsible for hypermethylation. So the general organization of the pathways is always the same, although the particular cofactors that are involved differ. And so from these and other studies, we think this provides very good evidence that there is really an instructive model initiated by the oncoprotein that's responsible for epigenetic silencing. A more recent piece of data we have that supports that idea, it came from this pathway over here. This is the pathway that mediates the CIMP phenotype in colorectal cancer cells that contain an activated BRAF oncoprotein. And it's shown over here. It involves four cofactors, MAPG, CHD8, BOC1, and DNMT3B. Now, this mutation, an activating BRAF mutation, is commonly found in melanoma. This is a point I will return to at the end of the talk. And so we asked, might this same pathway be responsible for epigenetic silencing in melanoma cells? And this just shows one piece of data that supports that. Here we're knocking down these four cofactors that we identified in colorectal cancer cells. We're analyzing expression across a panel of markers. These are CIMP genes. These are used to classify colorectal cancer tumors. We find that in this unrelated cancer type, a human melanoma cell, that these same genes are silenced, and virtually all of them become reactivated when we knock down these same factors. So here's a case of a oncoprotein-directed epigenetic silencing pathway that functions in two completely unrelated cancer types, and really we think highlights the importance of this mechanism in epigenetic silencing of tumor suppressor genes. Now, there's an important implication, we think, of the finding that oncoproteins can direct silencing of tumor suppressor genes, and that's illustrated over here. This is a working canonical model of how the RAS oncoprotein transforms cells. It's thought to do so through downstream signaling pathways like PI3 kinase AKT, which promotes cell survival, resistance to apoptosis, and through this MAP kinase pathway here that signals cell cycle progression. But what we found through these very same pathways, RAS silences tumor suppressor genes, and very similar for what we find with the BRAF pathway as well. And that really indicates that RAS and BRAF promote secondary oncogenic events by silencing these tumor suppressor genes. And we speculate that this is why BRAF and RAS are such potent oncoproteins and why they're found so frequently in human cancers. Some other things that we've learned from the screen is that it really takes a lot of work for the cell to keep these genes silenced, because they're these pathways often involving many components, and if you knock down any one of the components, that leads to reactivation of the gene. And so from a point of view of drug discovery, that's good. It indicates there are many potential targets that can be used to reactivate genes. So the epigenetically silenced state is stable, but it's not irreversible. And we've been able to reverse that with both biological and small molecule inhibitors. And those fall into two types. And I'll use as an example of that, this is a pathway we discovered by the RAS oncoprotein mediates the CIMP phenotype in colorectal cancer cells. The pivotal component in this pathway is a sequence-specific DNA binding protein called ZNF304. ZNF304 in normal cells is kept at very low levels because it's subject to polyubiquitination and proteasomal-mediated degradation. In RAS-transformed colorectal cancer cells, RAS leads to the inappropriate upregulation of two factors that mediate ZNF304 stability, USP28, a deubiquitinase, and PRKD1, a protein kinase that's a cofactor of this deubiquitinase. And so in RAS-transformed cells, ZNF304 levels increase. That causes binding of ZNF304 to promoters that it normally wouldn't occupy in normal cells, and recruitment of a co-repressor complex that leads to transcriptional silencing. So this is the kind of mechanism that explains why the gene is on in normal cells when ZNF304 levels are low, but off in RAS-transformed cells when ZNF304 levels inappropriately increase. Now, we can reactivate this gene not only with shRNAs, but with small molecules, and they can be broken into two classes. One is those we would call promoter proximal, that are bound to the promoter, such as 5-azacitidine, an inhibitor of DNA methyltransferase 1. But we can also reactivate the gene by targeting these upstream factors, such as PRKD1, with this small molecule inhibitor. And we couldn't have guessed PRKD1 would be involved in silencing. We could have only identified it because we did an unbiased large-scale loss-of-function screen. And we think in many cases these upstream regulators may provide more efficacious and specific targets than a molecule like 5-azacitidine. So I just want to finish this portion of the talk by saying that while we often see the sequence-specific DNA binding protein being inappropriately upregulated in cancer cells, leading to epigenetic silencing of tumor suppressor genes, we've also found cases where cofactors in the pathway become upregulated, and that leads to silencing. And I'll show you an example here. This is a pathway by which the RAS oncoprotein silences the fast tumor suppressor genes. One of the components in this pathway is TRIM37. TRIM37 is an E3 ubiquitin ligase, and we got very interested in it because it's located in the 17q23 chromosomal region, which is amplified in up to 40% of breast cancers. So we wanted to determine how TRIM37 functioned. It was bound to this epigenetically silenced fast gene, a chromatin mark that's very typical of epigenetically silenced genes is monoubiquitination of H2A. And we found that TRIM37 is a new H2A ubiquitin ligase. Here we're showing the ability of TRIM37 to monoubiquitinate H2A alone or in the nucleosome. Because this is amplified, because this is in an amplified region, the levels of TRIM37 in breast cancers that contain the 17q23 amplicon is increased, and we show this increased level of TRIM37, that TRIM37 at these increased levels binds to a well-known repressor complex called PRC2. Increased PRCA2 activity has been found in a number of cancers, in particular prostate cancers. And what we found is that when TRIM37 binds to PRC2, not only does it mediate H2A ubiquitination, but it actually changes the occupancy pattern of PRC2. So now it binds to and silences many genes that are normally unoccupied in cells that contain normal TRIM37 levels. So upregulation of multiple components in these pathways in different cancers can all result in epigenetic silencing and drive cancer progression. So I want to now turn to the second part of my talk, and that has to do with an oncogenic splicing factor. As I imagine everyone in the audience knows, splicing is a step that's involved in normal expression of genes. It involves the removal of introns and the ligation of exons. It's mediated by a large complex referred to as a spliceosome. And where this became cancer relevant is that there have been a number of studies, cancer genome sequencing studies, over the past 5 years that have found recurrent mutations in basal spliceosomal factors over here. And I think this is really one of the most surprising findings of cancer genome sequencing efforts. A lot of what we've learned from that we already knew or was largely predicted, but this was really a surprise. And we became particularly interested in this because one of the genes that becomes frequently recurrently mutated is a gene called U2AF35 or U2AF1, and it's a protein that we've worked on for a long time. And I'll just summarize what we know about it as shown over here. U2AF is a heterodimer. It's composed of a large subunit that recognizes a polypyrimidine track and a small subunit that recognizes the AG dinucleotide at the 3' end of introns. And it's really one of the first step in spliceosome formation, and therefore it's the subject of a number of regulators. And we've studied this protein for a number of years now. We originally identified its activity, purified it to homogeneity, and cloned the two subunits and worked out how it functioned. A lot of this work was done by Phil Zamore when Phil was a graduate student in my lab. Phil is now one of my colleagues at UMass. He's the chair of our RNA Therapeutics Institute, is really a leading researcher in RNAi and small RNAs. So we and others have been interested, why does mutations in this subunit lead to cancer progression? And there's an obvious hypothesis was that these mutations would cause some kind of alteration in U2AF activity that leads to altered alternative splicing that would either inactivate tumor suppressor genes or activate oncoproteins. And toward that end, there have been a number of papers that have been published in which the oncogenic U2AF35 variants have been introduced into cells and splicing patterns looked at by some kind of global profiling. And indeed, they find you can get alterations in splicing. But I felt these papers were unsatisfying because in no case could they link the alternative splicing event to the transformed phenotype. So we adopted an approach that differed in at least 3 ways from these previous studies. The first is, in none of these previous studies did they, in all the previous studies, they introduced oncogenic U2AF into cells at some arbitrary concentration and in some often arbitrary cell type. We sought to transform a cell with oncogenic U2AF35. That hadn't been done before, so that we can examine the consequences of any altered RNA processing event to transformation. So we've been able to transform several immortalized cell lines with oncogenic U2AF35. This is the S34F variant. It's the most common form. But then most of the experiments that we've done have been in BAF3 cells. This is an interleukin-3 dependent immortalized mouse bone marrow-derived pro-B cell. It's been used to detect oncogenic activity of a number of oncogenes, such as BCR-ABL. And when the cells become transformed, they enables IL-3 independent growth. So this is what one of our typical experiments look like. If you take BAF3 cells, parental cells, or where you ectopically express wild-type U2AF, you withdraw IL-3, all the cells die, you see no cell recovery. If you introduce BCR-ABL oncogene, you very quickly find IL-3 independent proliferation. But if you introduce oncogenic U2AF35, you also see transformed cells arise, although it takes a longer period of time. The cells, once they arise, have very similar growth rates, though, to the BRCA-ABL independent, to BCR-ABL transformed cells. And we can confirm that these cells are transformed because we can implant them into mice and they form tumors. So that's what we did, is we looked for RNA processing alterations in these oncogenic U2AF35 transformed cells, compared those to some parental normal cell. And here we did a second thing that was a little different than the other studies, because we knew that not only does U2AF affect splicing, it's also been shown to affect mRNA 3' end formation. And so we analyzed our RNA-seq data not just for alternative splice site use, but also for alternative use of cleavage and polyadenylation sites, CP sites, where the 3' end of the mRNA is. And so what we found is, like in many of the other studies, several hundred genes that undergo some form of alternative splicing. But the most frequently altered RNA processing event was alteration at the CP site, in particular increased distal CP site use. So we wanted to focus on this event. And so we took a number of the candidates that were identified by the global RNA profiling, and we confirmed that they had increased distal CP site use using qRT-PCR assay, where we're measuring a common site and then a site that's downstream of the normal polyadenylation site. And so you can see in all of these cases, we see increased use of the distal CP site. And then the third thing we did that's a little different, and we do this in all of our functional genomic studies, is very early, right after the screen, we try to identify particular candidates and follow that we think are going to be the most instructive, and follow those rather than following all several hundred genes. And the one we decided to focus on was ATG7. The reason we did so is that ATG7 encodes an essential autophagy factor. Autophagy is a context-dependent cancer prevention mechanism, and loss of several autophagy factors such as Beclin-1 and HEG5s have been shown to contribute to tumorigenesis. But we thought the most striking, the compelling reason to follow ATG7 is that ATG7 had been conditionally depleted in hematopoietic cells, and these mice have an autophagy defect and develop an MDS-like syndrome. And myelodysplastic syndrome is a disease in which U2AF mutations, U2AF35 mutations are most common. So for this audience, I'm going to focus mostly on the cancer-related aspects of this study and not the RNA processing, but I just want to show you, just want to summarize for you. We know the precise mechanism by which this mutation leads to use of the distal ATG7 CP site, and it has to do with a complex called CFIM. This is a complex that regulates use of alternative CP sites. It binds to the proximal CP site, and there are two forms of this complex that have antagonistic function. One of these contains a 59 kilodalton subunit, which increases use of proximal CP sites, and the second complex has a 68 kilodalton subunit instead, and that decreases use of proximal CP sites. And what we found is that in cells with wild-type U2AF35 that are not transformed, they directly recruit this 59 subunit form of the complex, leading to use of the proximal site. And in cells that bear this mutation, they are ineffective or less effective in recruiting the 59 complex. That leads to occupancy of the 68 complex and use of the distal CP site. And I just show you one piece of evidence in support of that idea. Here we're doing an RNA immunoprecipitation assay, and we're measuring binding of CFIM subunits to the proximal ATG7 CP site. And in non-transformed cells, you see they're predominantly bound by the 59 subunit, not the 68, whereas in transformed cells, you see a switch, and now there's predominant binding of the 68 kilodalton subunit. Okay, so why does use of the distal ATG7 site transform cells? So if you use a distal CP site, you'll have a long 3' UTR, and in many cases it's been shown that long 3' UTRs harbor translational repressive elements. So that caused us to look at ATG7 protein levels, and so here we're doing so by immunoblotting, and you can see in the control cells and in BCR-able transformed BAF3 cells, they contain much higher levels of ATG7 than in the than the U2AF35 transformed cells. And we could further confirm that this long 3' UTR is a translational repressive element because we can fuse it to a luciferase reporter gene and show that results in decreased luciferase activity. So a prediction of this idea is that that loss of ATG7, decreased ATG7 levels, is responsible for transformation. And so to confirm that idea, we knocked down ATG7 with an shRNA and looked to see if it can transform BAF3 cells. And very similar to what we found for introduction of oncogenic U2AF35, we can see the emergence of IL-3 independent growth with very similar kinetics to that we found with oncogenic U2AF35. And we can confirm that those cells are indeed transformed because they form tumors in mice. This predicts that the U2AF35 transformed cells have an autophagy defect. We can confirm that using well-established assays for autophagy. And here our ATG7 knockdown transformed cells provide appropriate positive controls because these are, by definition, essential autophagy factors. And so here we're looking at two markers that accumulate in cells that have defective autophagy, P62 and LC3B1. And you can see we see very similar accumulation of these markers in ATG7 knockdown cells and U2AF35 transformed cells. This is another assay for autophagy in which we're measuring autophagic flux using a dye called cyto-ID, and this is parental cells. You could see that we see a loss in autophagic flux when we knock down ATG7 or when we transform cells with U2AF35S34F. So why does autophagy, loss of autophagy, lead to transformation? One of the hallmarks of cells with an autophagy defect is they acquire mitochondrial dysfunction. One of the readouts of mitochondrial dysfunction is increased reactive oxygen species. And here what we're showing is that U2AF transformed cells and ATG7 knockdown transformed cells have increased levels of reactive oxygen species, here using this dye mitosox to measure that. Increased reactive oxygen species can cause mutations, so we predicted these cells would have a mutator phenotype. And here we're measuring the mutator phenotype by looking at the spontaneous mutation rate in two proteins, HPRT, selecting with a drug called 6-thioguanine, and the sodium-potassium ATP fixase, selecting for cells with mutations in this gene with Wabane. And so what this shows is that you get in the U2AF35 transformed cells and ATG7 knockdown transformed cells have increased mutations, spontaneous mutations in HPRT. And moreover, we can suppress those using the antioxidant ascorbate. And very similar results when we look for the rate of mutation of the sodium-potassium ATPase using the drug Wabane. So the final experiment I want to show you before I put these together into a model is we asked whether U2AF35S34F is required for both initiation or for both initiation and maintenance of the transformed phenotype. And we did that by transforming cells with the doxycycline inducible U2AF35S34F. We transformed cells but in the presence of doxycycline that allows expression of this protein, and after we obtain IL-3 independent cells, we remove doxycycline. That leads to the loss of this oncoprotein. That leads to the restoration of normal use of the ATG7 CP, proximal CP site. That leads to the reaccumulation of ATG7 to normal levels, but the cells remain transformed as evidenced by their ability to proliferate in the absence of IL-3. So we conclude from that experiment that this oncogenic U2AF variant is required only to initiate but not maintain transformation. And so we could put that together into a model that's shown over here. In normal cells that contain wild-type U2AF35, there's recruitment of a CFIM complex that contains the 59 kilodalton subunit, leads to use of the proximal CP site and normal levels of ATG7. Cells that have a mutation that contain this U2AF mutation now no longer efficiently recruit this form of the complex. Instead, there's a default binding of the CFIM68 form of the complex that leads to use of the distal CP site, resulting in a decreased ATG7 levels, defective autophagy, mitochondrial dysfunction, genomic stability, acquisition of secondary mutations that lead to the transformed phenotype. And we think that this explains why it takes longer to transform a cell with oncogenic U2AF35 compared to BCR-ABL, because it takes time to acquire the appropriate secondary mutations. And so just finally, I want to say that we think that this mechanism is not just what we have found in cell culture, but also is what occurs in patients. And so to come to that conclusion, we collaborated with a group at Columbia who has a large collection of samples from patients with myelodysplastic syndrome. And what we're doing here is comparing in bone marrow samples from these MDS patients use of the ATG7 proximal versus distal CP site in those cases in which have wild-type U2AF or contain the oncogenic form of U2AF. And you can see we see a statistically significant increased use of the distal CP site that is very similar to what we observed in our experiments. Finally, in many cases, MDS progresses to acute myelogenous leukemia. The oncogenic U2AF35 mutants have also been found in AML. And so to provide additional support for this, we did a bioinformatic analysis on the data from TCGA on AML samples. And again, we see a statistically significant increased use of the distal ATG7 site in patients that contain oncogenic U2AF35 compared to wild-type U2AF35. Okay, so the third part of my talk, I'll like to discuss a well-known anti-cancer prevention mechanism called oncogene-induced senescence and apoptosis. If you take a normal cell and you introduce into it an activated oncogene, the cell senses something that's wrong and mounts a response to prevent further progression to cancer. The response can either be the induction of apoptosis, cell suicide, or in many cases, a state of permanent growth arrest referred to cellular senescence. And this is now well established to be a fail-safe mechanism that prevents proliferation of cells at risk for neoplastic transformation. So we decided, and this is work that was really initiated by Narendra when he was in the laboratory, to study this process, and we decided to use as a model system the BRAF oncogene, which I've briefly talked about already. The BRAF oncogene encodes a serine threonine protein kinase. It's a downstream effector of RAS. It's signaled through this MAP kinase pathway, BRAF-MEK-ERK. BRAF is a proto-oncogene. To become an oncogene, it has to acquire an activating mutation. This predominantly happens at this one position, the V69E, which increases protein kinase activity and results in constitutive activity signaling through this pathway. The activating BRAF mutation is found in a number of solid tumors, but as I've mentioned already, it's most frequently seen in melanoma, where it occurs about 50 to 70% of the cases. Now, what made this such a good system to study oncogene-induced senescence is that there is a benign lesion called a mole or a nevus, and this is the same cell type that causes melanoma, namely a melanocyte. But unlike a melanoma, this is a growth-arrested lesion. The nevus can persist for decades. It has characteristic molecular features of senescence, and it contains in approximately 80% an activating BRAF mutation. So this is a case of a successful anti-cancer prevention mechanism, and this is a case where somehow that prevention mechanism has failed or been disabled. And so to try get insight into that, we wanted to identify factors that were required for oncogenic BRAF to induce senescence in melanocytes. So Narendra pioneered an shRNA screen, which is briefly outlined here, Basically what he did is he induced senescence in cells by introducing BRAF V600E, and then he looked for shRNAs that can overcome the senescence and allow the cell to grow. And we reasoned that such shRNAs would be directed against the factors that were required for BRAF to induce senescence. So this screen worked very well for him and allowed him to identify 17 factors required for BRAF-mediated senescence. And this just shows an example of how profound the senescence mechanism is. Here we take a small amount of cells, we plate them, they form a confluent lawn, but if we introduce a BRAF V600E, they become growth arrested. But if we knock down any of these 17 factors, they can still form a confluent lawn even in the presence of BRAF V600E. So what are these 17 factors? They're shown over here. 16 of 17 of them were what we expected. They're factors that are involved in transcription or signaling. 3 of the factors were well-established tumor suppressor genes, P53, MEN1, and BNIP3. And we would expect to find tumor suppressor genes in such a screen because this is an anti-cancer prevention mechanism. But what surprised us was one of these factors was a secreted protein called IGF-BP7, and it suggested that oncogene-induced senescence might occur by some kind of an autocrine-paracrine mechanism. So Narendra went on to study the role of IGF-BP7 in melanocytes, in nevi, and in melanoma, and I'll briefly summarize what he found. It's shown over here. In normal melanocytes, and we think in many other normal cells, IGF-BP7 is transcribed and secreted at a low basal level. It operates through this autocrine-paracrine mechanism, and it actually inhibits BRAF-MEK signaling. It's downstream, its transcription is downstream of BRAF-MEK signaling, and that establishes a negative feedback loop, which we think dampens both BRAF-MEK signaling and in proliferation in melanocytes and other cell types. So what happens if this cell acquires an activating mutation in BRAF? That leads to a large increase in signaling through these pathways, a large increase in synthesis and secretion of IGF-BP7, which induces senescence by 2 mechanisms. One is it inhibits signaling through this proliferative pathway, and second, it activates a group of senescent genes that we identified in the primary screen that really enforce the senescent phenotype. So this is an example of a successful tumor suppressor mechanism. The cell has an oncogenic mutation but doesn't go on to form a tumor. To form a tumor, this mechanism has to be disabled, and we found that in many cases it's disabled because IGF-BP7 itself becomes epigenetically silenced. However, these cells remain exquisitely sensitive to IGF-BP7. So if we introduce IGF-BP7 exogenously by, for example, providing the recombinant protein, this winds up efficiently inducing apoptosis in these cells for 2 reasons. One is it inhibits BRAF-MEK signaling, and these cells are dependent or addicted to signaling through this pathway. This is the rationale for using inhibitors against BRAF V600E or MEK, pharmacological inhibitors. In addition, IGF-BP7 activates a dedicated apoptotic program shown over here involving genes that we isolated in the primary screen. So this experiment that I've now summarized is really an example of a well-established concept in cancer biology. That's namely that if you have a cell that's missing a tumor suppressor gene, if you're able to restore it, you'll have a beneficial effect. So for example, there have been hundreds, if not more, papers published that have shown that if you reintroduce a tumor suppressor gene into a cell in culture, you'll induce growth arrest or apoptosis. And there have been probably a few dozen papers that have taken that to the next level and shown that if you reintroduce a tumor suppressor gene in tumors in engineered mouse models, you'll attenuate tumor growth. And in fact, this has prompted clinical trials in which venerable tumor suppressor genes such as P53 or BRCA1 are attempted to be introduced into tumors using some kind of a gene therapy approach. And while this it conceptually makes a lot of sense, from a technical point of view, it's very challenging because most tumor suppressor genes are intracellular proteins, so you have to get the DNA into the cell, and it's cancer, so you have to get it into 99.9% of the cells. But for a secreted tumor suppressor, it's much easier because all you really have to do is get the protein, not the DNA, and all you have to do is get it to the outside of the cell. So this gave us reason to believe that IGF-BP7 and perhaps other secreted tumor suppressors could be therapeutically useful. So at that time, Narendra did the following experiment to establish that concept. He took human melanoma cell lines that can either contained an activating BRAF mutation or were wild type for the RAF-RAS allele, introduced them into the flanks of mice, and when tumors reached a certain size, we injected either PBS or recombinant IGF-BP7 into the tail vein and monitored tumor growth. And what he was able to show is that IGF-BP7 had a very strong attenuation of tumor growth, and that was specific for the melanoma cell lines containing the activating BRAF mutations. So that's by way of introduction. What I want to tell you now is we have developed another way in which we could introduce IGF-BP7 and other tumor suppressor genes into cells, and that's using adeno-associated virus vectors. This is work that we've done in collaboration with Guangping Gao. We wouldn't have done the work if Guangping Gao weren't at UMass, and he's head of our gene therapy center. AAV has really revolutionized now approaches to gene therapy. It's a small DNA virus. It does not integrate. The transgene persists as an episome. It allows for long-term expression of the transgene for decades. And unlike some other viruses, like adenovirus, for example, it's relatively non-immunogenic. So in our experiments, the target cell we're using is the muscle cell. We introduce an AAV derivative that expresses IGF-BP7 into the muscle, and we turn the muscle into an IGF-BP7 basic biomanufacturing facility. It's secreted from the muscle and then encounters the tumor where it has its effect. So this is the kind of experiment we do, very similar to the one I showed you before, with the one difference is now, rather than introducing IGF-BP7 recombinant through tail vein injection, what we do is introduce it into the contralateral anterior tibialis muscle. And so what you can see here, we're using that same cell line, SKMEL, which contains activated BRAF, and we have this very strong anti-tumor effect. This shows an example of another BRAF-positive melanoma cell line where AAV9 delivered IGF-BP7 has a very strong anti-tumor effect. This is a very aggressive cell line. You could see that in the control animals we have to sacrifice after 17 days to remain compliant with our IACUC protocol, whereas you could see tumor suppression all the way out throughout the time course of the experiment. And this is in case another cell line that we found was sensitive to IGF-BP7. It's a colorectal cancer cell line that actually contains an activating KRAS mutation, which is upstream of BRAF. Now, one of the things we're continuing to do with Guangping is to optimize the delivery platform, and particularly we're trying to get it so that IGF-BP7 is expressed only at the local site of injection. And that would enable us to be able to remove, to get rid of IGF-BP7 and AAV if desirable by a simple muscle biopsy. The AAV9 is considered to be a muscle-tropic AAV variant. However, AAV9 has and other AAV variants have the undesirable properties that they can transcytose across blood vessels and enter the bloodstream. So you see over here where we're looking at the distribution of AAV9 using a luciferase reporter gene, And with the standard AAV9 controls, we get a lot of expression at the site of injection, but we also see a lot of expression elsewhere, and this is predominantly at the liver where AAV9 accumulates once it gets into the bloodstream. Guangping has developed another much more muscle-specific variant that's shown over here. You could see now that when we inject AAV9 here, we don't see the liver distribution that we saw with wild-type AAV9. We've removed the anterior tibialis muscle, and after a week, what we see is still large amounts of expression in the control AAV9, but much reduced expression in this AAV9 variant, and that's quantitated over here. This is the control AAV9. It actually increases after myectomy, whereas this variant over here decreases and is much lower levels. Another way we can look at viral distribution is we can look at viral genomes in the muscle. You could see with both wild type and the new variant that Guangping made, in the injected muscle, there's lots of AAV9 genomes, but you could see with wild-type AAV9, we also see a lot of genomes in the contralateral anterior tibialis muscles, which is greatly reduced when we use this new variant. And so this final slide on this subject shows that we haven't lost any of the anti-tumor suppressor activity by delivering IGF-BP7 using this viral vector. So I just want to finish the talk by saying that we found IGF-BP7 really serendipitously. We were actually surprised. We didn't expect to see a secreted tumor suppressor protein. But now that we've seen it and studied it, we think that there might be a lot of therapeutic potential. So we've now developed methods to proactively go after and to identify new secreted tumor suppressor proteins. And our approach is shown over here. We first just scrutinize available data sets for genes that are down-regulated in cancer versus normal tissues. These are putative tumor suppressor genes. We select those that encode predicted secreted proteins. We clone and express those genes and look first for their ability to kill cancer cell lines, but not non-transformed cells. And I'll show you one example where that seems to have been quite successful. This is SRPX. It had the two properties we were looking for. We interrogated expression profiling databases and found that SRPX is downregulated in lung cancer and other solid tumors, and it has a predicted signal sequence. Here we're actually confirming that prediction by analyzing by QRT-PCR SRPX expression levels in a series of adenocarcinoma and squamous cell carcinoma samples, and you could see that in many cases there's a large decrease in SRPX expression relative to normal lung. Here we're looking, confirming that SRPX has a predicted signal sequence. We're expressing SRPX in cells, 293 cells, which don't express the endogenous protein. You could see we see some SRPX in the lysate, but we also see some in the conditioned medium, indicating that some of it's being secreted. And if we remove the first 35 amino acids of the protein that contain the putative signal sequence, we no longer see secretion into the conditioned medium. This shows that recombinant SRPX can efficiently kill a variety of lung cancer cells that are in the NCI 60 panel. By contrast, it does not kill non-transformed cell lines like immortalized human bronchial epithelial cells or MRC lung fibroblasts. And so here we're looking at SRPX delivered by an AAV9 vector to suppress tumor growth. Here we're seeing suppression of tumor growth of A549 lung cancer cells. Here we're seeing another suppression of another cell line, HOP92, colorectal cancer cells, including colo205 and HCT116. So we're excited about this approach. It's based upon a natural cancer prevention mechanism. We're restoring a protein that's been lost in a cancer cell, not introducing an exogenous foreign agent. We think it's applicable to many cancers. We're going to find secreted tumor suppressor genes From a drug development point of view, this is a very rapid approach. Most cancer drug development programs first validate a target and then look for inhibitors against that target. Here, this approach gives us a first-line therapeutic. And then finally, based on gene therapy studies, we think the future drug development steps are well established. So I'll just end. I want to acknowledge the people that are involved in this work. This is an oncoprotein-directed tumor suppressor gene silencing. This is a problem, as I said, we've been working on for a decade now, so there's a lot of people involved. Narinder was involved in some of the important early work, but the people who are most recently involved in these studies are Ryan Serra, a former graduate student in the lab. He's now a postdoc with Ollie Randall at UMass, and Mingying Fang, who is a current postdoc in the lab. Oncogenic splicing factor directed transformation. This has primarily been carried out by Sung-Mi Park, a postdoc in the lab. We had a very important collaboration with a group from Columbia who runs this myelodysplastic syndrome clinic. That's headed up by Azra Raza. An important part of that project is also Siddhartha Mukherjee. Some of you may recognize him because he is the author of the book, The Emperor of All Maladies, The Natural History of Cancer. If you haven't read the book, I highly recommend it. And then finally, oncoprotein-directed senescence apoptosis. This work was initiated by Narinder when he was in the laboratory. Most of the experiments I talked about today with AAV have been done by Sanchita Bhatnagar. She's a former postdoc in the lab. As of this fall, she took an independent faculty position in the Department of Biochemistry at the University of Virginia. And as I said, we would not have done these experiments if it were not for my colleague Guangping Gao, who's head of the UMass Center for Gene Therapy. And I thank you for your attention and be happy to take any questions.